Welcome everybody to the session on community ecology, diversity of species and traits. And we're going to kick off with Sarah Cousins, our first speaker, from the grasslands. Coming up.
Okay, thank you. I'm glad to see so many here with tough competition from the Ilka session. I'm a professor in physical geography, and I've been working with landscape ecology and particularly plant community diversity and community change for a long time, mainly using historical maps to investigate landscape changes and the effect on plant diversity. Let's do this. Move. Next slide. Yeah, I did. Well, let's do next.
Slow down a bit.
Oh, yeah. No, I think the batteries. Oh, I can still talk, maybe.
Give it a minute.
This will be over for 2 minutes. Just enter.
Start again.
I can do next. Okay. Right. Okay. So usually we use historical maps from maybe 17th century to look at grassland changes, and we know that it's obviously one of the world's most species-rich habitats, and we know that a long continuity of management, low-intensive management, had created a buildup of species. On the other hand, about 90% in the place where I work in Sweden, 90% of the grasslands have been lost during the last 100 years, so that's obviously a great concern. I usually get questions, and one of my postdoc actually was asked that yesterday, what about the grasslands before people started to use them, or before we had these maps? And there's also a big question on where these species were in the landscape before the grassland management and the people started to manage them. Now. Right? Works. So I wanted to look into, or try to explain, where were the grasslands before man came here and started to manage them. Scandinavia has been under ice. So 22,000 years ago, it was the last glacial maximum, pressing the whole landscape down. And then 10,000 years ago, you can see to the left. Right, left. You can see the map or the red circle show where I work. And to the right, in that case, that land, how it looked like 6,500 years ago. And you can see that the whole landscape was submerged underwater. So the water level here, where I work, is 160 meters below present day. So we wanted to look into how much land came up out of the water, and particularly how much potential grasslands that have emerged during the last 4,000 years. We picked 4,000 years because that's where we have the oldest records of grassland management from pollen records. So we wanted really to look at this, don't use these guys, this area. So the land uplift is today about 1.2 millimeters per year, or no, 20 to 30 centimeters per 100 years. That's easier to explain. So it's quite a substantial uplift even today as we work here, and we can see it on maps. But further back, the rebound was much quicker. So we're talking about almost several centimeters every year that actually came out of the water. So if you have a fairly flat landscape, it's a substantial amount of area that's coming up. So we picked a landscape model, and we used landscape scale data, a digital tool terrain model of 23,000 square kilometers to look at land uplift together with quaternary geologists. And we picked out the areas where we thought could be potential grasslands. So a very flat slope, so 1 to 2 degree slopes. And then we also wanted to look at management. How much species diversity do we have in shore meadows today, to try and explain the effect. And first, to pick the shore meadows, we have a map here, an old historical map from 1901. See if this pointer works. No. No, it's actually, this is fun. So the green stuff on the map is the old shore meadows. So these were used to collect hay. The pink, yeah. The pink is arable fields, and the white is grazed outfields or forest. We don't really know, because these maps were made to really show how much could you tax the farmer for arable fields and meadows. To the right, you have an image, a satellite image today, and if I had a pointer, you could point out some of these meadows that have actually disappeared. They become more encroached with trees. So we picked out meadows that were managed 100 years ago and compared it to meadows that, this is a meadow that's still managed. And if they're not managed, they look like this. It's basically more or less reeds. So first of all, we did this landscape model. And this is a short movie, should start, showing the land, potential grasslands coming out of the water every 100 years. So the blacker the landscape, that is where you have fairly flat slopes with grassland management, possibly grassland management after people come in. So this starts at 4,000 years ago, and then it moves every 100 years over. The red stars is sample sites. So this is just a small fraction of the area we worked in. It's basically, so you will be able to see it. This is like the freak, the nightmare presentation. What else can happen? So, slowly, land is coming up, people moving in this landscape. As soon as the water disappears or the land is coming up, we have evidence of really old ancient remains of hunter-gatherers from a long time ago. The red line is the sort of shoreline today. So now the film has stopped, and this is what could be potential grasslands, and we have only fractions today. So in our landscape model, we could see that almost 8,000 square kilometers land emerge out of this. More than 2,000 square kilometers are potential meadow. So about 50 to 150 square kilometers coming up every 100 years. And as you can see from this image, it's from the same place. And the man standing in the top, you can see almost like a small island. So that's actually the same area as this small woodland up there. So even in 60 years, this is quite substantial changes. And we can imagine that the farm, when people started to farm this, this was land and really useful for grassland management and to collect hay or have grazing animals. So, sample design for looking into species diversity today. We use a borderline, I think you can see it. Here you have a slight undulating border between the grassland that is affected by the salty water. Remember that the Baltic is quite brackish. And the area to here that's drier. So you can actually use a line, which is the border. And we place sample sites from the border and out towards the sea and up towards the forest today. And the Baltic has no tide at all, because it's such an inlet area. So it was significant change or significant species richness in the unmanaged grasslands compared to the managed, also at both at plot level and at site level. And also, really interesting is that we can see that the species richness actually accumulates after the land, or the further away from the sea, which we take as age up towards the forest, we can see that species start to accumulate. However, the management is such a strong effect with the animals walking around, spreading species, so we can't really say, the interaction is not significant. But plot age is important. Right. So today we have only 10 square kilometer managed shoreline meadow today. It's only less than 0.04% of this whole area. So there are only really fractions of grasslands left, these typical shore meadows. And we have shown in this study that beside these ideas that the old ancient grasslands before man came or management started could be calcareous uplands, floodplains, still they're very limited in space. But these grasslands, they would have been well connected, huge amount of areas all over the world where we have this isostatic rebound. So this is sort of created a gradual and continuous creation of large and really well-connected grasslands. And obviously, if this area was destroyed with sea level rises, which we can almost certainly foresee, the whole grasslands that you can see on these images will be squeezed together between the forest and the land uplift, which makes it really important for management and conservation today how to manage for rising sea levels. At the same time, you have this rebound. I also wanted to point out that this landscape that is rebounding and the archipelago that we'll hear more about in the next talk is a really exciting area because you can actually date the islands, how old they are, or the areas. So in our small plots along the grassland, we can almost for certainly date when it came out of the water, or areas on the islands in the archipelago. So it's a really cool study area. And that was it. Well.
We have some time for some questions, which you probably want to feel given the little technical pitch.
Yeah, I got too nervous when everything went wrong, so I spoke even quicker than usual.
That's okay.
Do you have any information on any large animals that were grazing these areas at the time?
I mean, we don't have information. We know that there were large grazers in the area. I mean, all the uroks or whatever. So we know that they were there. But we can never pinpoint exactly because these grasslands are actually moving up, so they become forest. But we can model. I mean, you would be able to say that they were grazed, but we don't know. It's just potential grasslands. Yes.
So you were talking about plant species. Now, do you have any information on invasive species and others in
Invasive species.
Invertebrates.
Invertebrates. No, I don't. But there was a talk earlier this morning about invertebrates, but in the same system. But we haven't looked into this, so you're welcome. They're still there. So far, as long as we don't have any sea level rises.
That's it? Sorry, one really quick one.
Yeah, quick one. I was wondering, you were talking about climate change and basically sea level rise as a threat. Are there other threats to?
Yeah, management, lack of management. Yeah. So I mean, it's from other studies in the same area. We know there's only 2% grassland left. So management is the really important, and obviously they become so fragmented. So it's only dots here and there. So that's the largest threat at the moment.
We're going to move on to our next speaker, staying safe with the other sessions. Thank you very much, Sarah. We're now going to move on to John or Jan. Talking about biodiversity making spatial and temporal dispersal on fragmented semi-natural grasslands.
There's the point.
And that's left and right. you get a new one?
No, I'm going to go for it. I'm going to give it a try. Okay, thank you very much. Welcome everybody. After this amazing talk from my former postdoc supervisor, I'm going to talk about something we've been doing in pretty much the same, at least in the same system. So, in case the first thing is going to, this should come to nobody as no surprise that global change today is threatening global biodiversity. And, well, habitat fragmentation is one of the most pervasive factors. It's widely recognized. And plants are particularly hard hit because, of course, they're sessile organisms. When there's substantial changes or disturbances to the environment, they just can't run away. Well, why is that? Or if we want to rephrase the question, what is actually the thing which is causing these, what drive plant diversity in fragmented landscapes? And again, it's pretty widely acknowledged that dispersal processes in our fragmented landscapes is one of the key aspects. And if we look at what is very widely studied and very intensively studied is specifically how dispersal limitation in space is a problem for plants, because in the most broadly speaking terms, plants just are poor dispersers. But that is not what I'm going to talk about today. To make things absolutely clear, I'm going to talk about dispersal maintenance, like the opposite. Because if you look, now why would we do that? If you look at meta-community theory, it actually explicitly states that spatial dispersal not just limits, but it also maintains diversity. And if you think about it, in a fragmented landscape, you also have many species around. And in theory, the species which are maintained are completely different and independent from the species which may be limited by dispersal. That's what the theory. And mostly how we assess limitation is by sowing seeds in a site where plants are absent, and if they grow, if they manage to establish, we assume that they are dispersal limited. Now, to establish how dispersal maintains diversity, we would ideally exclude dispersal from a system and observe which species go extinct. And then we could say these species are maintained by dispersal. Of course, that's not that easy to do. But beyond the mere spatial aspect, we also have in the meta-community theory, there's also talk about temporal dispersal as being an important aspect, which is also important for maintenance of diversity. There are, via clonality and perenniation, you have remnant populations which can cling on for some time, but you also have persistent soil seed banks, which are important aspects for maintaining population demographics. They are biodiversity reservoirs in fragmented landscapes, and they might potentially buffer against losses of biodiversity above ground due to disturbances. Now, most of spatial and temporal dispersal are usually studied separately and independently from one another, and there's only very few studies which combine the two aspects. And not at all in a spatially explicit context like with habitat fragmentation. And that's weird because the few studies which do address it come to estimates about that 40 to 70% of diversity is actually maintained by their combined action, and we have a whole body of literature focusing on the 30% species which are dispersal limited. So that's I found that a bit odd. But the main problem is that there's a big gap of empirical data supporting that this is actually the case. But as I mentioned before, there's quite some empirical and practical complexities with excluding dispersal from a system and observing which species go extinct, and might not be something we actually really want to do. And trying to do is look how dispersal maintains it is really of importance to understand, both fundamentally and of implied ecological importance on how we should manage our fragmented landscapes. So I asked myself three basic questions. So if we lose spatial dispersal from a system, would this mean that the fragmented plant communities, that they would disassemble because diversity is declining and species are being lost? The turnover in the plant community as a result of that, is that deterministically? Is that non-randomly? Is it non-random, and is it driven by the potential of species to spatially or temporally disperse? And finally, if we lose spatial dispersal, can this decline be buffered by the fact that there's temporal dispersal in your system via either seeds or clonal and perennial mechanisms in plants. So the study area, I pretty much worked in a similar system. But so as I mentioned earlier, it's not easy to discern how temporal and spatial components of dispersal maintain local diversity. So we basically need a unique kind of system in which we know, perhaps, where spatial dispersal, or at least some part of it, has been removed from the system. Now, Sarah elaborately talked about it. We had an opportunity in the Stockholm archipelago. It's like some additional information. As of current, it's about 24,000 islands, ranging from big permanently inhabited islets to rocky skerries. They hug the coast of Stockholm here. At their widest point, it's about 60 kilometers wide and 150 kilometers long. And it has a long history of small-scale agriculture and grazing. And today, it's mostly what's left is very highly fragmented grasslands in a sea and forest matrix. But where did we have the opportunity to study this dispersal maintenance? Well, in the archipelago, it meant often that farmers had to physically move their cows between islands during the summer. So they had the home island where the farm was, and then during the summer, from early spring to late autumn, they moved the cows between islands for sheep. And that's been a common practice throughout the archipelago. But we have these where it's still done today, and for the most part, it's like this, that it's been removed from the system. So we actually have a system where we know that the green landscape infrastructure is similar between these. So we have the similar levels of background dispersal, if you want to call it like that. But a substantial component of the man-made spatial dispersal by moving the cows has been removed from the system. And it should point out that these grasslands here are still managed to precisely prevent forest encroachment. So we're talking about still managed grasslands in both systems. So what we did is we have this here is the still rotationally grazed one. And here rotational grazing has been removed from the system. So we inventoried 77 plots where we looked at both all the plant species above ground, and we took soil samples to also look at all the species which were beneath in the soil seed bank to quantify the temporal dispersal in each site. So I will not bore you to death with the analysis, but basically for the first research question, we simply did GLMs where we decomposed the alpha and beta diversity metrics. And we looked at how landscape metrics and rotational grazing had an effect. Then we looked at specifically the grass, the herb layer, and are these species being specifically driven extinct by spatial and temporal dispersal potential, for which we did a nestedness analysis, which we extracted information on the positions of the plots and species and related them to those characteristics to identify what was driving the extinctions. And we did this again, this nested analysis, and with inserting the seed bank's data to understand whether temporal dispersal would affect the nestedness pattern, and we would consider that a buffering effect if that was the case. Essentially, we found, if you look at the landscape infrastructure, the structural connectivity, if you like, was very weak effects. But at the loss of rotational grazing, at alpha diversity, these are three, the names have gone here, but these are three types of grassland species, like specialists towards generalists. This is the seed bank below ground and the herb layer above ground, and you always see a very strong effect of the presence of, these ones have the presence of the networks. So if the networks were abandoned, we see that many species go extinct. Beta diversity, we have exactly the opposite pattern, where beta diversity increases. So there's an increased community dissimilarity because species are randomly or non-randomly disappearing from your system on the abandoned grasslands. And this was true for both the above ground communities and the below ground communities. If we then looked at community turnover, again, we saw very limited effects of the structural connectivity in the landscape. But there was again a strong impact of the fact that the grazing networks were still intact, yes or no, where there was a strong, significant nestedness pattern, which you see here, where we calculated this no overlap decreasing fill metric. And we extracted the plot ranks and species ranks to study further what was actually determining this nested pattern. That this was nested means that it's deterministic and predictable, the species which are going extinct. And it's very much related again. So the position of the plot here is related to whether they're still grazing, yes or no. And the higher the number, the less species and the more species have gone extinct compared to the grazing networks. So which species are going extinct? It was not related to basic dispersal mechanisms like but it was related to the potential of animal dispersal. And that was based on whether there was really actually seeds been found or recorded in poo in other studies, for example. No, it wasn't related to that in general. But if you looked at it decomposed on specific species groups, specialists were very hard hit by the disappearance of the cows. And that maybe not make sense, but it does make sense because we know that cattle or sheep and cows are very, very good dispersers, and even specialists benefit from it if they have no specific adaptations for being dispersed. Is it dependent on temporal dispersal mechanisms? It was strongly related, like clonal species, which across all species, if you had no clonal potential, you were much more prone to extinction. And this was again true specifically for habitat specialists which had no possibility for clonal persistence or temporal dispersal. But then if we looked at the soil seed bank specifically, this was even actually the best predictor of presence in the system. So the more a species occurred in the seed bank, the less likely it was going to go extinct throughout the system. If we then looked at the temporal buffering effect, if we redid the nestedness analysis, inserting the species occurrences in the seed bank, would it offset those species being lost above ground in the herb layer? And there we found no relation with the grazing network, which was a bad thing. But we did find that the shift in the nested matrices was related to island size, which basically implies that on the islands where the rotational grazing had disappeared, if it was a big island, there's still some potential of species being reintroduced from the seed bank if we would restart management. But unfortunately, it's probably limited in space and time, the effect that might still last. So what does it mean? If we lose spatial dispersal, it's deterministic community assembly by non-random species extinction, mainly of non-clonal and non-seed bank species among specifically habitat specialists. And it appears serially aligned those effects. So if you look at the abandoned grazement, it looks that today diversity is limited by dispersal, but maintained by temporal dispersal because it's preventing further community disassembly. In the grazing network itself, we have a huge and massive effect of the presence of the grazing network, having high diversity, but also homogenizing the communities. So what I want you to take home is that I have gathered some empirical data showing that spatial dispersal is a key agent in maintaining local diversity. But unfortunately, today we are all well aware that non-connected grasslands are the norm. So maybe potentially temporal dispersal may be a very or more important agent than temporal or spatial dispersal. And the good thing is that it's a major management opportunity that grazing networks appear so effective. Then that was it. I will thank you very much for your attention, and I have any questions either now or later on if you want to talk to me. Thank you.
One quick question.
Are you sure it's dispersal that's resulting in species being there or not being there? So if it's a grassland, it's a disturbance maintained system. So the lack of grazing means that you're getting a transition from a healthy ecosystem to one where a few tall species of grass have come to dominate. There's lots of more abundant material. So is it not simply that you're ending up with essentially a degraded grassland because there's no fire or grazing?
No. Because it's the grasslands on the abandoned network are still managed, either by stationary grazing, so that people bring in cattle in the beginning of the summer, or that they slaughter them for meat at the end of the summer, or they're being mown. So they're all still being managed, yeah.
Okay, I think we better move on to keep on track. Thank you very much.
Yeah,
We're now
you're
going to hear from Inika talking about niche differentiation of plant species along an NP gradient in Eurasian grasslands.
Okay, this is the pointer. Can I use this one? Will it
Yeah,
work?
yeah,
Okay.
and then
Just
just,
go.
hold the pointer down when you want to use it. It should be fine.
I'm going to give it a try then. There you go. So I hope you can all hear me. Okay. So good afternoon. My name is Ineke Roeling. I work at the Copernicus Institute of Sustainable Development at Utrecht University in the Netherlands. And I worked together with my colleagues from the Copernicus, but also with a colleague from the Radboud University. And today I want to tell you that we found that there is niche differentiation of plant species along a nitrogen-phosphorus gradient in Eurasian grasslands. So, as you know, we have a problem with eutrophication due to agriculture, industry and transportation. We have an extra addition of nitrogen and phosphorus to our systems. And just to give you an idea of the scale, at the left here you see a picture of the exceedance of the critical load for nitrogen. And this is a picture of 2004. Only the light blue parts here have no exceedance of the critical level. Then at the right, you see a phosphorus addition to the system in the form of manure. That's not the only way. You also have mineral fertilizers and, of course, other additions. So just to give you, again, a basic idea of how bad it basically is. It's getting better. We have European policies in place, but it will take years before it's really reduced, and also phosphorus adheres to the soil complex. So this will be actually quite a legacy that we have. So what does it do? As you probably know, eutrophication in itself leads to a decline in biodiversity. Basically, you have a few species that grow very quickly and outcompete the slow-growing species for light. So only the fast growers stay in your system. But you can also end up with a shift in the relative availability of nitrogen to phosphorus. And we call this relative availability the nitrogen-phosphorus ratio. And as you can see here, it's actually a gradient with, at the low NP ratios, nitrogen limitation, then a very short part on the gradient where nitrogen and phosphorus are both limiting plant growth. And then you have a very large part along the gradient where you have phosphorus limitation, going all the way up to extreme phosphorus limitation. So what is this NP ratio? It is not measured in soil, it's measured in above ground biomass. As you can see on the picture, what you just do is you cut a piece of vegetation, you dry it, you take it to the laboratory, and you measure nitrogen and phosphorus contents. It is an integrative measure of the uptake of the vegetation during the growing season. To do so, you of course have to measure it at the peak of the growing season. And as I said before, and I'm pressing this point, it is a measure for the relative availability of nitrogen and phosphorus. Many studies have been done. They have invented basically this nitrogen-phosphorus ratio and have also looked into thresholds. And using field fertilization experiments, they found that nitrogen limitation occurs below an NP ratio of 13.5. Between 13.5 and 16, you'll have co-limitation, and above 16, your vegetation will be P-limited. So why should we care? Because it's important for diversity productivity patterns. So just presenting you some results from a study done by Martin Wasse and colleagues in 2005. They analyzed 270 plus sites of Eurasian wet grasslands, and as you can see here, you have the biomass production on the x-axis and the amount of species in a plot on the y-axis. And then you get this quite famous hump-shaped envelope, basically. What is important is that this envelope is different depending on whether your site is nitrogen limited or phosphorus limited. As we can see here, the red dots are the phosphorus limited sites. The curve is much narrower. and the peak is also earlier than the peak for the nitrogen-limited sites. What this implies is that if you have a phosphorus-limited site and you have eutrophication and probably an addition of biomass, it is quite likely that your species richness will decline much faster than if you would be in a nitrogen-limited system. Also, they looked specifically at endangered species, and they found that, as you can see here again, both the percentage and the absolute amount of red list species is higher for phosphorus-limited sites. So these phosphorus-limited sites are actually very, very important. Then we had another colleague, Yuki Fujita, and she looked into community trait means and how they change along the nitrogen-phosphorus gradient. And what she realized is that there is likely an interaction between the nitrogen-phosphorus gradient and productivity. So what she did is she removed the effect of productivity first. So what I'm showing you here are the unique effects of the nitrogen-phosphorus ratio on community trait means. So what do we see? Again, here at the left, and here as well, you have nitrogen limitation. And we see that the communities that are nitrogen limited have a faster growth rate and a high investment in sexual reproduction. Very easy to see. We have a high specific leaf area, we have a low leaf dry matter content, a high seed number, and a low investment in vegetative reproduction. Well, actually for the phosphorus limited sites, as you can see, the pattern is exactly the opposite. So just to summarize very quickly, we've seen 3 studies basically focusing on the community level. We've seen that the MP ratio is important for diversity productivity patterns, the maximum amount of endangered species that you can have in a system, and also that there are very big differences in community mean traits. This gave us the idea that it is very likely that if there are such big differences between these nitrogen-limited sites and phosphorus-limited sites, that there are likely different species growing in one place compared to the other place. So we asked ourselves, what is the relationship between the nitrogen-phosphorus ratio and the occurrence of endangered species? So we have 3 hypotheses, basically, that I'm going to show you today. The first one is that the nitrogen-phosphorus ratio explains part of the species composition and that it does so independent from other environmental variables. That species will have their own niche along the nitrogen-phosphorus gradient, and that the endangered species are likely to have smaller niches, which would make them vulnerable for changes in the nitrogen-phosphorus ratio due to eutrophication. So we used the same database that Yuki Fujita used. We excluded the plots that were close to the seaside or were too salty, or plots that were too woody. Ended up with 644 plots across 8 countries. A nice gradient also in the MP ratio when you are covering all of these different countries. And we only used species that occurred at least 10 times in our data set, excluding quite a lot. So we ended up with about 180 non-endangered species and 90 endangered species. The data set had, of course, information on the nitrogen, phosphorus, and potassium contents above ground, species absence and presence data, and also the mowing frequency. We also wanted to know, as I said, if the MP ratio has an effect independently from other environmental variables. Unfortunately, these were not included in the database, so we used Allenberg indicator values for moisture, pH, salinity, temperature, and light. So the first finding is that the MP ratio does explain part of the variation in the species composition. We used multivariate analyses, DCA and CCA, and this picture is hopefully readable at the back. Every green dot is a species. The species that are closer together often occur together in the field, and the red arrows then indicate the environmental variables. So what you will see here is a very clear effect of moisture. So here you will have wet sites, and here you will have more dry sites. Also, for the people familiar with DCA, we see that for the first axis, moisture is most important, and for the second DCA axis, MP ratio, very long arrow, and phosphorus implant are the most important variables. So the length of these arrows and also the results of the CCA, which I'm not presenting here, actually show us that the MP ratio explains an important part of the species composition, and that it is as important as salinity, soil moisture, soil pH, etc. It's actually more important than biomass, which we found very interesting. Then going along, this picture shows you the niche of plant species along the nitrogen-phosphorus gradient. And as you can see, we clearly see that species have their own niche. If you want details on how we calculated this, please talk to me later. The black dots in every bar indicate the mean niche width. The height of the bar indicates the niche width itself. So these are the niche optima, and these are the niche widths. And basically, you will see that there is a clear turnover of species when you are moving from nitrogen limitation in this part of the graph all the way up to phosphorus limitation in this part of the graph. The green bars are non-endangered species, and the red bars are the endangered species. So again, you will see, which is quite logical, we use the same database, that especially under phosphorus limitation, you will have a high occurrence of endangered species. And also, just take a look at the, as I said before, at the height of the bars, because what we were asking ourselves is there actually a difference in niche width between endangered and non-endangered species? And as I said before, we expected the endangered species to have smaller niche widths. So what we did is we looked at the species with an optimum under N limitation and for species with an optimum under P limitation. For N limitation, I can be very quick. There's basically no difference in the niche width of endangered and non-endangered species. And for phosphorus limitation, it's exactly the opposite of what we expected. So we basically see, and you can also see it in the previous graph, that we have quite some species with a very narrow niche, but we also have some endangered species which actually cross over to the boundary to nitrogen limitation. So yeah, why? Well, we are still thinking about this, but we have the idea that some of the species with a very broad niche that are still endangered may actually be more influenced by soil moisture or maybe soil pH, and that the MP ratio for them is not the most important factor to be in an environment. And this is why we want to know. This is a beautiful picture of the nature reserve, the Drentse A. As you can see, rare orchids, rattle, buttercup, and all kinds of different grasses. Basically, we just want to understand why a species is growing, where it is growing, and also how we can keep them. In the Netherlands, we have quite some problems. Despite all the management efforts, in some areas we are successful, in others we are not. So what we are saying is a species where it's growing depends on moisture, depends on pH, but it also depends on the nutrient limitation type. Thank you.
One quick question.
Do you have an explanation why your niche width is getting larger under phosphorus limitation?
As I said before, not exactly. We are thinking that for these species, other factors may be more important. Of course, you have species that are very, susceptible to changes in pH. And maybe these are species that are susceptible to pH, but not so much to the MP gradient. So that would be a guess. Another option is that these species grow in certain areas, especially in Poland and Siberia, where the nutrient amount available is so low that even with slight changes in nutrients, you get maybe quite a big shift in the nutrient-phosphorus ratios. That does mean that they are exclusively growing in phosphorus-limited systems, but that they can still have quite a large niche just because they're bound to these systems due to other reasons.
Okay, thank you. I think we better move on. Thanks for
If you have other questions, just ask me later.
So we're going to move on with
Yep.
Natural co-selection of plant and soil communities shape the biodiversity ecosystem functional relationship.
Maybe.
Do you know how to get the technician?
And then you have to see if it works.
Yeah, that's the pointer anyway.
Yeah, that works. So that's the main thing.
It's a PDF. Yeah, how do you do a PDF?
Somewhere in the background. Seems.
Will it work with this? I should do. Let me just try.
Yeah. Hello everyone, welcome to my talk. I'm going to present something that we've been working on for a few years now in our group on how community selection can increase biodiversity effects in grasslands. I'm from the University of Zurich, and I've done this study, or not only me, but a whole bunch of people have been doing this all together. So it's well known now that diversity enhances productivity. We've heard it also today a few times. In this study, in the biodepth experiments, the scientists showed that with increasing species richness, also the observed yields or productivity in the system increases. What was also shown is that actually this positive biodiversity effect, how we call it, can increase with time. So in this other study from the Cedar Creek biodiversity experiment, they showed that again with planted richness, so increasing species richness, the yield increases, but that positive slope increases with time. So in the beginning of the experiment, these are these more reddish curves. It is a positive effect, but the slope is more flat. And then towards the end of the experiment, actually the biodiversity effect is much stronger. So clearly there is something happening over time, and it's conceivable that there are evolutionary or selective mechanisms behind this. So when we then look into kind of this community selective effects, we have to actually go switch to other organisms such as bacteria, or also studies have been done in plankton, because they are much easier to manipulate, they have shorter generation times. So a lot of research on community selection has been done in bacteria. So in this study, what they had is they sampled bacteria from tree holes and grew them in the lab in 3 different mediums and experimentally evolved them over time. And also in bacteria, we can see this positive biodiversity effect. With increasing species richness, productivity of the bacteria increases. And then over time, actually these slopes can change, or just the overall productivity can either decrease or increase, or depending on the medium also very little happens. So our question was if we can actually extend these principles also to plant communities. To answer this question, we made use of a very large biodiversity experiment from Germany, the Jena experiment. It was established almost 15 years ago in 2002 out of a pool of 60 European grassland species. Every one of these rectangles represents a plot with a different species composition and also different species diversity levels, ranging from monocultures up to 60 species. And then within these main plots, main experimental plots of the main Jena experiment, what we did is in 2011, we established the Swiss boxes. They are the Swiss boxes because it was a Swiss research team messing around the German experimental field. So this is how these Swiss boxes look like. They were set up with a pool of 52 grassland species in a diversity gradient from 1 up to 8 species. And we did not only install 1 plot, but actually 48 of them all over the field. So what's inside the plots? I'm going to walk you through the experimental setup. So we have 2 aspects to it, the plant community history, so the above ground, and a soil community history. So for the plant side, we created 2 different selection histories. So selected communities and unselected, or old and new. To create these communities with selected plants, actually all of the plants were excavated, then brought to Zurich, seed were collected, and then we propagated seedlings in a glass house and planted these back into these plots in Jena. For the new plants, or for the unselected histories, we bought seeds from a company. Also,
made seedlings in the same kind of glass house and also planted them back into the plots. So in every plot we have old and new plants growing next to each other in two different kinds of soil treatments, in either co-selected soil, so this is soil that has been in the Jena experiment over all this time together with this specific plant community, and then neutral soil, which is a soil that has no common history together with the plants. So this soil was also excavated, treated, in some cases gamma sterilized, and then put back into the plots. Then we did lots of measurements in the field, so we collected, we clipped the grass, or like all the species above ground, then also sorted them, weighed the community biomass, and dried it first, and then weighed it to get above ground productivity. And we did this in 4 springs, so in 2012, 13, 14, and 2015. With this kind of data, we made this diversity productivity curves, as I've shown you in the very beginning, with plant species richness on the x-axis and productivity on the y-axis. And we did these curves for 4 years, so for each of the years that we sampled, and also for either the plant history or the soil history, so selected versus unselected communities, and kind of co-selected and neutral soil. Yeah. So these are the results for the above ground selection. So what we found is the community selection generally increases biomass. So these old plant communities generally were more productive. But what we also found is that this community selection treatment actually increased the biodiversity effect. So especially when we go from monocultures to 2 species, these slopes here are much steeper than they are here for the unselected plant communities. Because we wanted to see how the ancestors, basically, of these selected plant communities were doing, we had a look at the data of the main experimental plot right after the installment of the main Jena experiment. So we also looked at the years we looked at years from 2003 to 2004, so the first 4 years of the experiment, and we found that actually this plot here looks more similar to this one on the right, so to the unselected plant communities, than to this plot of the selected plant communities, because again, when you go from 1 to 2 species here, this slope is actually quite flat. So it seems that basically the ancestors in the beginning of their selective process, they perform similarly than to this new unselected treatments that we have in the field. It would be possible that all what we see here is driven by just 1 single or maybe 2 species. So we had also a look at the individual biomass for every species separately. And this is just a plot showing that if the value is basically above 0, then it means in the selected treatments or the old plants produced more biomass. And if the value is below 0, it means that the new plants produce more biomass. And this is data across all soils and across all years and diversity levels. And we can see that for the majority of species, actually, they had the biomass, a higher biomass in the selected treatment. So it seemed like several, or at least the majority of species, are actually driving what we see in the field. So this is the effect then of the soil history on the, again, on the above ground community. So on the left-hand side is the co-selected soil, and on the right-hand side is the neutral soil, also over the 4 years. And we find, oops, sorry, wrong way. We found that this kind of soil legacy or co-selected soil generally decreased biomass. So biomass is lower in this treatment, but it also somehow dampened the variation between the years. So here there's much more temporal variation. So we think that maybe the, in this case, somehow the interactions between the above and the below ground communities are well established, while here everything is still kind of trying to settle and the interactions aren't yet kind of fixed. So the mechanisms that we think are behind what we see here, we think that it might have been a sorting out from standing genetic variation. We know that when the experiment was set up, we had quite a large genetic variation. So with this selective effect, that there was just a sorting out of genotypes that are performing the best in this kind of communities. We also think that selection might not only have changed species performance, so how much biomass it produces, but also interactions between the species to really kind of optimally function in their specific community. The unselected plant communities could not catch up within these 4 years. So these selective forces do not primarily act during seed establishment and growth in the very beginning, but this seems to be a process that goes on over a longer time. This has some implications for the way we do research. So if we already see this kind of effects after 9 years of selection, what we had now in our experiments, and we can assume that natural communities have decades of coexistence or even hundreds of years, and that it would make sense to do this kind of experiments at a community level and not look at species in isolation. And this also, of course, has implications for conservation, because it might be that local species extinctions cannot be so easily made reversible and not be so easily reversed. Because if you have a local species extinction and then you bring just a species back into the community, but maybe from a seed bank or from some completely different community, that it actually might not be able to replace that specific, or like the species that got extinct. So it would make sense to protect species within their community context. So rather protect them in a national park or kind of in ecosystems instead of just freezing them individually in seed banks somewhere in the Arctic. So because only like this you can also protect the species interactions. And it also makes sense to conserve genetic resources as communities of species. So with this, I would like to thank my many co-authors, the helpers in the field, technicians who also helped the URPP Global Change and Biodiversity, and my research group. And I thank you for your attention, and I wish you a happy Christmas coming up soon.
Okay, really interesting talk. We have time for some questions.
Yeah?
Do you have any idea what some plant species do first? So selected plant species do first?
That's a good question. You have to ask them. No, I don't know.
Any speculation?
I would have to look at so we looked at the functional groups, and I don't know if you saw it, but there's not like a specific functional group, for example, doing bursts. So it's really a bit all over the place. Some are grasses, some are legumes. So there isn't like really a clear pattern. So I'm not familiar with all the life history traits of all the species. So sorry. Sophie?
So did you have the opportunity to calculate overyielding indices?
Sorry?
So you didn't keep the species also in monocultures?
Yeah.
Did you?
No, we had the monocultures and the mixtures.
Did you try to calculate any indices of indicate overyielding?
I mean, not for this specific study, but in the framework of the N experiment, I think we have seen overyielding quite often. Yeah.
I think there's random rules with the monocultures mixing both things. Okay, are there any more quick questions, or did I one really quick one?
Okay, no, sorry, I missed that part of it. Did you sterilize all the soil, or just for some of the treatments?
Only for some of the treatments. So actually, the soil was taken out, about 2/3 or half of it was sterilized, and then to create this neutral treatment, but the other half was the same soil that basically was already in the field, to have these two kinds of soils.
Okay, we better move on. Thank you very much.
Thank you.
So our next speaker, Chelsea Shields, who's going to speak to us about functional traits show variable predictability of tree growth across climate states and Norwegian forests.
Can you do it on here? Oh, great, excellent. That's the pointer. I'll just grab your chair for you.
Oh, thank you.
Perfect. All right. Hi, everyone. I'm Chelsea Chisholm, and I'm a PhD student at the Center for Macroecology, Evolution, and Climate, based in Copenhagen. And I'm going to talk today about some work I've been doing with a number of collaborators. Ooh, thank you. Also, I'm not lazy. I just have an injury, so if I need to, I'm going to use this chair. So don't judge me. I'm going to talk to you about some work I've been doing on modeling growth responses of forest communities using a long-term data set of 30 years in Norway. And in particular, incorporating functional traits and trying to use those to predict growth response of these trees. So we know that there's a number of drivers that are changing communities today. Human-driven land use, for example, as well as climate change. And forests are a really great spot in order to examine some of these impacts because we can actually track back through time, given there's a number of inventories that are available in various countries, where we can look at, for example, changes in growth rate or fitness of these trees over time, and look at what drivers are pushing those, and then being able to use that to predict into the future. So there's a number of abiotic factors that can be important here, such as climate, precipitation, and temperature. But there's also a number of biotic factors that we're interested in as well. So what is driving competition within these forest communities? So if we can think about a focal individual of a particular size, this focal individual is surrounded by a number of different neighbors. And we can think about competition in this network in terms of just the number of stems around this focal individual. We can think of it in terms of the number of con versus heterospecifics. And we can also think about it in terms of how large those trees are and what distance those trees are from that focal individual, and sort of creating a neighborhood around this particular tree. The other aspect that we can include, which I'm interested in, is the functional traits, whether dissimilarity in functional traits actually matters for these trees and their growth rate. So for example, if we look at two very similar trees, they might be very functionally similar and there's a lot of overlap in terms of specific traits. When we look at that compared to a tree that may be a little bit more different in terms of leaf or wood traits, they might not overlap as much. So there's all these aspects of sort of spatial distance and size of neighbors, as well as their functional identity that might have an impact on the competitive effect of those neighbors on that focal individual tree. So there's a number of ways in which we can think about how traits might actually impact this tree in terms of competition. So Mayfield and Levine produced this paper that nicely kind of summarized coexistence theory in terms of two different axes of functional traits. So for example, we can think about the bottom axes here, niche difference, just how dissimilar you are in terms of trait values. So sort of like the absolute value of difference in terms of those functional traits between you and your neighbor. But we can also think about competitive ability difference, where we don't want to have so much of an asymmetry in terms of functional traits that confer competitive ability, such that you have exclusion, competitive exclusion of a partner. So there's this sort of zone of coexistence in here that we can look at. And we can use functional traits to try and get at these two different aspects. So this has been done in forestry research before. Kunstler et al. Looked at these niche differences as well as relative fitness differences as competitive ability difference in a forest inventory based in France. And he actually found support for relative fitness differences or competitive ability, explaining species interactions in these particular forest plots. So given this background, we wanted to ask a few questions. We wanted to look at what processes have been leading to the current structure of boreal forests, and what might be changing their species composition into the future, in particular focusing on functional traits and if they help us predict these particular responses. So there's two main questions I want to address for this talk. One of them is there any evidence for stabilizing niche differences, so this niche dissimilarity in these particular forest plots versus the relative fitness differences or differences in competitive ability in tree growth across species, and does climate mediate any of these competitive effects? So to do this, I've been using a forest inventory that is based out of the Norwegian Institute of Bioeconomy Research. It's this fantastic 30-year forest inventory that has, after pruning for some more managed environments, 330,000 records of 4 to 5 measured diameter at breast height for each focal individual from 1986 to 2015. This is in permanent plots that are spread in a 3 by 3 kilometer grid in the south and a 5 by 5 kilometer grid in the north across the entirety of Norway. And for this particular work, we focused on 7 species, which make up approximately 95% of the coverage. across Norway. So it's a very low diversity system as well. We also gathered functional trait data together. So there was measured specific leaf area, functional traits that we took within the plots in a subset of plots across a latitudinal gradient. And then we also took wood density from TRI because we weren't able to measure these within the plots. And these two functional traits confer something about sort of the strategies that these species take, more acquisitive, fast-growing strategy versus a more slow-growing, stress-tolerant strategy. So we used Bayesian hierarchical models in order to model this growth rate. And essentially, this is the equation right here. Essentially what we're predicting growth rate with is with size of tree. So the bigger you are, the faster you grow, as well as growing degree days and precipitation. And we also incorporated a crowding index, and this is where we pull together the functional trait information that we're interested in. So this crowding index is essentially a summation of the size of the neighbors and the relative distance to that focal tree. And then we also have a lambda parameter that we can then vary and incorporate functional traits within. So we have 5 separate models that we ran where there's a null model where lambda is 0. So essentially competition doesn't matter at all. Model 2 is a neutral model where we're assuming that the competitive effect of all species is equivalent on that focal individual. Then we have a model 3 where we estimated competitive effects of species separately. And then we had 4 separate models where we incorporated the trait data. And this is where we get at that niche dissimilarity versus competitive ability differences. So we can look at the absolute differences between these two traits, SLA and wood density. And that's the stabilizing niche differences between species, so that they're different enough to coexist. And then we can also look at relative fitness differences or hierarchical trait differences between species, where we just look at the actual differences between the two species, the focal individual and its neighbor. And then we can sum that across, and that becomes our crowding index for these species. So just to go into the results, what we found is that precipitation, temperature, and tree size all positively influence growth rate. When we looked at the actual trait values, both of these two models 4 and 5, we actually found that traits don't really matter across most of our species apart from one where there's a positive influence of wood density within aspen trees. So in effect, we found that traits don't really matter for competition within these Norwegian forest communities, which is very different from what other papers have found. For example, Kunstler in the more diverse French inventory plots, and also within some of the larger meta-analyses that he's done recently in 2016 that have looked across a number of different biomes. So in taking that, we also found that the neutral model was our best model. We examined this using something called the WAIC, which is like a AIC information criterion. So the best model came out as a neutral model, where the competitive effect is equivalent. There's no difference in terms of the species, in terms of competitive effect on a focal individual. So then taking this, we decided to reformulate this, and using some of Kunstler's previous work that said that SLA and SSD are both important in directly predicting growth, we just used our neutral model and we incorporated SLA and SSD wood density into our growth rate model. And then we took this data and we binned it across a number of different climate bins. So this is where we wanted to look at how climate might mediate the effects of competition across all of these different communities. So we binned it into low, mid, and high temperature, and then low, mid, and high precipitation and the combinations of those. So if we look at the effect of competition across all of these different plots, we can see here in the bottom is lower temperature, it goes up to higher temperature. That in general, competition becomes more important in more, let's say, benign environments, which is what we would expect. And then if we take a look at the actual effect of traits across all of these different bins as well, we see something that's a little bit different. So we find that trait values aren't really so important in the more benign environments, and that potentially there's some effect of traits, which are both positive in the harsher environments, low precipitation and low temperature. And part of this might be due to some sort of trade-off in terms of competition, where we're having high competition in the higher areas, and then once we've kind of filtered our species into those that can establish in these more harsh environments, then we find that these differences in trait values start to matter. So our question originally was, are biotic interactions an important component of how forests will respond? We found that yes, but our interactions were not so much mediated by functional traits. We found they were less important in this system than previously found. Our trait effects do differ with climate, with both SLA and wood density having an effect at low temperature. However, we do bring in sort of a caveat that there might be individual variation that's important. When we bin this data, we're essentially taking the trait value, a mean trait value for that particular bin, and ascribing it to each of these individuals based on a per species average. But there might be some, potentially in these low diversity systems, niche spread that we're not really capturing that individual traits might be able to tell us more about this. So that's something that's important that we might be pursuing in the future. So within Norwegian forest communities, I think we can still use these trait values in order to predict what climate change might do to these communities in the future. For example, if some of these more low temperature, low precipitation areas start to warm, competition might become more important in those actual communities, and they might start selecting for species with particular traits. For example, those which have higher SLA values. And then we might start to see some change in community composition as this occurs. So with that, I'd just like to finish my talk and thank everyone who's involved with this, in particular NIBIO for being so welcoming to a Canadian-Danish researcher, and all my funding bodies and collaborators as well. Thanks. Yeah.
Any questions for Chelsea? We have time for a few.
Yes.
Thanks very much. That was really interesting. I'm just wondering about these traits that you chose to use, the SLA
Yeah.
and the density.
Yeah.
Obviously, they're really frequently used traits and they have advantages. Do you think there are any other traits where there would be a wider range between those 7 species?
Definitely. And this is a limitation of this study in using just 2 separate traits. Sure, they're very important, but there's a suite of other traits that we could be using. And there's been previous research not necessarily in trees, but in annual plants. So some experiments that have been run by Jonathan Levine and Oscar Godoy, where they looked at sort of suites, and Nathan Kraft as well. They looked at suites of traits and showed that niche dissimilarity is better predicted by sort of a whole suite of traits versus just single traits. So that might be why we're not actually picking up any signal of that at all as well. That's certainly a limitation. Exactly, yeah. And so the wood density varies a bit, but you're right. There's not so much variation in terms of these traits. Yeah.
So following up on that question, I was just wondering, what do you think is the most limited resource that's important for the competitive advantages of the tree species? Because depending on whether it's like water or light or whatever, the functional traits might be more from the ground.
I think it really does depend on where they are, right? So these environmental filters might be different depending on these bins. And Norway's unique as well because, I mean, it gets a ton of water, right? So we are binning based on climate, but that low precipitation areas might be a lot of water compared to, for example, a desert area. So I don't see that as being potentially a limiting factor. So it is sort of a unique environment in which to address this. But it is a low diversity system, and I mean, those types of systems cover vast areas of the world as well. So I think it's interesting to also look at this, not just in tropical environments where a lot of this research happens as well, but also in low diversity environments where there's many monocultures where we can actually try and tease apart how these species or these individuals are coexisting. Yeah.
Which species do you think from your work would be the winners under a sort of warmer, wetter system?
Under a warmer, wetter system, we might start to see some of the hardwoods coming up, for example. There's actually, well, right now there's this interesting aspect of Norway in that the expansion of spruce is actually still occurring down southward instead of northward as well. So potentially we might see some of them being outcompeted or something like that. But there's also successional issues too, where, I mean, if something's already been established, then it'd be very difficult for other species to come in then too, like given a late stage succession, for example, for other species to come in and infiltrate those communities. So, yeah.
Quick one. I was actually going to ask very quickly to just take advantage of your position here. Had you picked up, you just mentioned species range shifts that you picked up in this 30-year data set.
Mm-hmm.
Is that something you used to pick your functional traits, or are they sort of buried in the functional traits? Or can we'd see?
Species range shifts.
Like, is the small species pool, is it changing from over the last 30 years?
No, it's not changing very much. Growth rates are changing within plots, for example, but I think the species ranges aren't shifting a huge amount. It's a pretty slow system, but yeah.
Great, thank you very much. Our next speaker, Lynn, is going to talk to us called Looking for Determinants of Growth Rate in 300 plus perennial plants.
Thank you, everyone. Thank you for coming to my topic. And doesn't work. Okay, thank you. So today my topic is about try to dig out what could the determinants for growth rates. Growth rates, as we all know, it will show the plant, it will grow fast or slowly. Then it will affect the plant population and the community dynamic. And then it's very necessary to find what is behind the growth rate. What could be the driver for growth rates? Many studies shows that relative growth rates is related with specific leaf area. But most of the study comes from the seedlings of woody species or annual species. Then what about the growth rates for perennial plants? And also, what about the grades for the plants during the whole growth season? Whether these leaf-specific traits or genome size or the volume storage organ will affect the growth rates. And if we consider the phylogenetic information, whether their relationship will change. Also, the environment condition will have an impact on growth rate or not, whether they have direct impact or indirect impact through other traits. So first of all, I track the foliage growth rates in the botanic garden for about 300 perennial herbs. Why in botanic garden? Because it will be time-consuming to collect about 300 species in the natural field. While in the botanic garden, the species grow in their optimal habitats. And it's easier to track the 300 species during the whole growth season. And for each species, I measure the stretched foliage length. And here is the final result. I use the data to track the growth trajectory for each of the species, and show here. This is the Julian from the January the 1st, and this is the leaf foliage length. We're using the logistic function and show here. K means predict maximal foliage length. And B, we call the standardized growth rate. GR is absolutely growth rate, showing the slope of the tangent here. The dot here means the peak growth. And the standardized growth rate is the absolute growth rate divided by the K. So this is the main difference or different meaning for the two types of growth rates. The absolutely growth phase are constrained by the size of the plant. Means the plant, if it's bigger, then they will have higher absolute growth rate. But for the standardized growth rate, we can compare the plant without considering the constraints from plant size. Then we collect other traits, like specific leaf area from Lambda database. And then the genome size collected from our cursor. And the size of a storage organ, we calculate from the picture of a clonal plot database. For example, shown here. And the calculation is based on the size of the storage organ, for example, here or here. Then we wonder what are their relationship between the growth rate and other trees. From here, we can say that specific leaf area is not related with growth rate. And the genome size is significantly related with growth rate, means plant with big genome size, they will grow faster. What about standardized growth rate? We got a similar result. And also, we found that the plant with bigger volume of underground storage, they will grow relatively fast. And it's understandable that there is no difference here, but there are obvious relationship here because of the K. The second question, whether there's phylogenetic conservatism here. First of all, we get the phylogenetic information from Duffin phylogenetic database. From there, we use lambda as the phylogenetic signal. Then we use the phylogenetic generalized least square model. Lambda was calculated use the maximum likelihood method. In order to compare, or in order to show the phylogenetic effects, we also assume lambda is 0.1 as non-phylogenetic regression. So here's the result. The relationship between absolutely growth rate and other trees, non-phylogenetic and phylogenetic regression. From here, we can say the relationship between GR and the genome size was significant when we not consider phylogenetic. But the relationship, the significance disappears when we consider phylogenetic information, which shows the absolutely growth traits at the genome size and their relationship shared the phylogenetic conserved, which means they shared the evolution history. Also, for the standardized growth rates, we got the similar results. What about the rule of environmental factors? So we use Ellenberg value as the environmental factors. Here we named light optimal because the Ellenberg value means collect the data of environment condition for the species which occurs mostly there. And then we use the structural equation model to get the result. And this is the p-value for the model, which is almost 1, showing the model predict well. If the two pairized, if these two pair trees, their relationship is p-value is bigger than smaller than 0.1, then I show the arrow and the coefficients here. From here, we can say the nutrients optim have indirect effects on growth rates, meaning the plant in which prefer rich habitats, they will supporting a big volume size and then have absolutely growth rates. What about the standardized growth rates? We got the same result. Besides, we also find the direct effects on standardized growth rates, which means, but it's negative, which means the plant which prefer rich habitats, they have relative slowly growth. It is because in high nutrient habitats, they will compete badly, or the similar situation happened for light. Okay, here's the conclusion. First of all, their relationship. We find in our result, the plant with big genome size or with big volume, they grow relatively fast and absolutely fast. So plant with big genome size, they grow faster, probably because in the early of the growth season, they divide their cells first and then inflammate the cell later. And the plant with bigger storage organ, they could grow faster by supporting the resource from the organs. So we've also found the phylogenetic conservatism here, both in the growth trees and other functional trees, and also their correlations. And the environment effects. We found the direct and indirect environment facts for the growth trees, showing the species which prefer growing rich habitats or growing open habitats. They have relative lower growth rate or standardized lower growth rate. But at the same time, they have a larger volume which could support their fast growth. And from all the study, we can say what exactly happened during the whole seasonal growth. This is the peak growth. Before that, at the beginning of the growth trees, what is the driver of the growth trees? The trees which can mobilize the resource for fast growth, like the storage organ. And after the peak growth, then the trees about leaf economic trees will start to work for the resource accumulation. Therefore, most of my result shows the specific leaf area does not related with two types of growth rates. Thank you. Thanks for my. The whole part of the part of the whole leaves.
So the
all leaf structure.
Total surface area
yeah.
of the plant.
Yeah.
So every 2 weeks you went and measured the surface area on every one of the plants. That's a lot of work.
Yeah. I mean, every plant I will stretch the leaf from the bottom to the top part. Measure them once every 2 weeks and track for the 300 species every 2 weeks until the end, like September.
Great, thank you.
Okay. Any comments?
People nodding in agreement with you.
But it's very impressive the amount of work that you've done.
Yeah, it's a small part of the result. Thank you.
So we might just wait for a minute or so for actually where is ready?
Yeah, I'm ready.
Okay. So next speaker, Sophie's going to talk about partitioning phytoplankton community variation using 18S rRNA sequencing and worker species data.
Okay, hello everyone. So I'm going to be talking about a completely different, not completely different group of organisms, but they're quite more primitive than plants, and they are unicellular. So what the focus of this talk is basically how the information that we take out of two very different species identification techniques, and namely the molecular one and the morphological one. Can we actually, do they give us different indications when answering ecological questions? For example, those that have to do with assembly processes of phytoplankton and what are the drivers of these assembly processes. And when I refer to assembly processes, the two main theories about this are the neutral and the niche. So in the neutral theory, organisms, different species are thought to be ecologically equivalent, and therefore the presence or absence of species from a given patch is determined by dispersal limitations. So how well connected are the patches between them in a meta-community framework. Whereas in the niche approach, we have local abiotic factors that drive the establishment of species and their populations at a certain patch, which of course is determined by the own life history traits of the species. So this was the first aim of this study, to basically investigate what is the relative role of these niche or neutral theories, processes in shaping phytoplankton assemblages in marine environments. And to address this question, we actually had a really cool data set that was derived from the Ecology project that was run in Greece a couple of years ago. And the field part of this project encompassed 7 gulfs across the Aegean Sea, with 5 stations in each gulf, sampled at 3 different depths. So we had 105 samples, so observations, and these gulfs were quite different between them because they represented a eutrophication gradient, and we actually have a lot of information on the physical environment, so the dissolving inorganic nitrogen and phosphorus, temperature and salinity that we all know affect phytoplankton growth, and also the diversity and composition of species. And most interestingly, the community was expressed both microscopically and so in the microscopic observations, we used sedimentation and filtering, and we did a rigorous taxonomic analysis. But we also used Illumina sequencing of partial 18S RNA genes. And then we assigned the operational taxonomic units to genera that are in GeneBank at the 97% similarity level. So for both of these approaches, we had 105 samples, and we had either genus abundances or genus read data, respectively. So this naturally creates the question of whether these two approaches of species identification give us the same indication, answer this ecological question in the same way, or do they provide different indications? And it is natural to ask this question, particularly if we actually see the results from these two taxonomic identification approaches, because they see the phytoplankton world in very different ways. And the molecular approach in particular sees many different groups that the morphological approach cannot see. For example, it can see ochrophytes, haptophytes, and chlorophytes, as well as other groups, whereas the morphological approach can detect, is more sensitive to diatoms and dinoflagellate species. So these groups that are not detected in the morphological approach might be because they are quite small and they are very difficult to identify taxonomically. But this different sensitivity also reflects the different compositional dissimilarity between our samples. So this grouping reflects the molecular and this grouping the morphological data, and here you can see the pairwise comparisons of our 105 samples based on their genus abundances. So we have a lot of variation to explain, but the patterns are quite different within each method, which makes this question even more relevant. And it is relevant to also ask whether the environment is affecting assembly processes, because also we have a lot of variation in our abiotic variables such as dissolved inorganic nitrogen, phosphorus, salinity, and temperature amongst our different gulfs. But we also have this really cool information about connectivity of our gulfs, and this was actually quite different between pairwise comparisons of gulfs. We established this information through a tracer simulation experiment whereby a parcel of water is released in a gulf, and we measured its probability to go into a different gulf based on the hydrodynamic circulation of the Aegean Sea, which is mostly governed by surface winds rather than thermohaline circulation. So we have all this information that can help us address our questions. And now the data analysis approaches that we employed were two multivariate eigenvector approaches. The first one is called PCNN, and basically it explains the variation in our community data set based on our set of known environmental variables, but also a set of unknown variables that we didn't really measure, we didn't account for them. And how do we come up with this matrix? Basically, this method tries to find spatial structures based on our community data set that are either fine scale or broad scale, and assigns them into different PCNN variables. And the cool part of this approach is that with a redundancy analysis, we can then partition the variation in our community on the environment, the unknown variables, into the unknown part, and in the overlap that we always want to minimize because we don't want any covariance between the two. And the second approach, which is the IM approach, basically we have this extra component of connectivity that is weighing these spatial structures, these unknown variables. And again, the procedure that follows is exactly the same as I described before. So we hypothesize that the PCNM analysis will basically show us whether the environment plays a role in structuring phytoplankton assemblages. But then, in the IM analysis, which adds this extra component of connectivity, if this manages to explain more variation in our community variation, then this means that the role of dispersal might be important as well, pointing to neutral theory. Which was not the case, however, because in this IM approach, we actually, when this component of connectivity was added, the explained deviance was either less or similar between the two approaches. Therefore, we do not see any evidence for improved explained variation when we account for dispersal limitation between our gulfs. Another thing here, we see the importance of the environment and the spatial structures between the different identification techniques. So basically, in the molecular data, which are shown with the red dots, we see that this effect of connectivity is consistently not important between the two approaches. And we also see that the morphological approach, in the morphological data, the environment is less important, whereas spatial structures that we didn't really account for are more important. And more particularly, in the morphological data, we see two spatial structures, namely these PCNM1 and PCNM2 variables. One is fine scale, one is broad scale, and also the role of salinity being quite important, as seen by their coefficient values. Whereas in the molecular data, they seem to be, the community seems to be driven more by the dissolved inorganic nitrogen and temperature. Which brings the question whether the taxa that we record microscopically could be more susceptible to processes that we didn't really measure, like grazing. We are actually counting these samples, measuring these samples now in the lab of zooplankton biomass, but were hidden within these two variables. And on the other hand, taxonomic groups that were seen by the molecular approach, such as the ochrophytes, the haptophytes, and the chlorophytes, could they be more sensitive to drivers such as dissolved inorganic nitrogen, which tends to be limiting during the summer in the Aegean coastal waters, or temperature? And to conclude, we basically saw that local environmental processes seem to be driving phytoplankton assembly more than the effect of connectivity between the different gulfs. Molecular and morphological data seem to convey different information, both with respect to biodiversity and compositional trends that they express, but also with respect to the drivers of these patterns. And it seems that these two approaches that we employed in this study, the PCNM and the AEM approaches, can be promising tools in answering these kinds of questions, the relative role of neutral and niche processes. And yes, I think that one of the next steps for this approach would be to also test these approaches with presence-absence data rather than general abundance data. And with that, I would like to thank all the team that took part in this Ecological project, and thank you all for listening.
Time for questions? You're looking very tired. Don't take that. Okay, it was really high one.
Yeah, I had a question basically about one of the last slides that you showed. Yeah, this one. Because it's clear that when you have your molecular analysis and morphological analysis, I mean, here you see certain species you can see visually and others you cannot. They are maybe too small or too difficult to identify under a microscope. So with a molecular analysis, you see more genera, more species. You were also saying like maybe some of those, like chlorophytes, are more influenced by the DIN and by the temperature.
That is a hypothesis.
That's a hypothesis.
I need
yeah.
to see
no,
that.
No. I understand. I think that your data suggests this quite. Well, the thing I what is actually the question? The question is, I mean, you have you have the same samples, no?
Yes, they're exactly mirroring each other. They come from the same bucket. It's just two different approaches.
But then, how did you connect your different approaches? Maybe I didn't get it because we're tired. But I mean, you have molecular data, morphological data, but still your same measurements that you took, I think, in the field, basically in the water, are completely different.
So these are trying to explain the community that was based on the morphological data.
Yeah.
Of course, the environmental drivers were the same,
yeah, exactly.
But the community is depicted by two different approaches. Here, it's just the data that we recorded when we were watching under the microscope,
yeah.
And here they came out from the molecular approach. So, because they have these two completely different ways of seeing the phytoplankton world, The drivers seem also to be different. And it's quite puzzling because based on whichever approach you select to use, you might have a completely different interpretation of what are the drivers affecting phytoplankton assembly. To us,
yeah, I think we're going to move on to the next speaker. Thank you very much. Okay, so next speaker, Simon Ford, is going to talk to us about trip filtering, about vegetation density underlies contrasting patterns of phylogenetic and functional diversity in temperate forests. Back into the forests, but looking at animals.
Thanks. Thanks for the kind introduction. Looking to your faces, I will try to make it short, and I'm pleasured that I have a lot of photos actually in my talk. So there's still a lot of debate about the loss of biodiversity, the loss of functional and phylogenetic diversity, the loss of genetic diversity, and there's still a lot of question marks and the threat that cause actually threats to biodiversity, as you can see, for instance, here. And what comes out of this conceptual framework and what's also coming out of this session here is that one of the main reasons because there is remaining question marks is because it's very hard just to measure a phylogenetic diversity or a functional diversity of a species assemblage just by walking through the forest or just by walking around. And that's so hard because patterns of functional and phylogenetic diversity typically do not actually match those of species diversity. So you often have a specific numbers, but you have much more less or more phylogenetic functional diversity than you would expect just by these numbers. One example for this comes from a French group where they model like the species numbers on the the species numbers here on the left-hand side, the phylogenetic diversity here in the middle, and the residuals of both here. So if we say that functional diversity could be explained only by species numbers, we would expect only white maps here. But in fact, there is blue and red color, which means we have areas where there's much lower phylogenetic diversity than we would expect by the species numbers, or higher phylogenetic diversity. There's the red color here. And the explanation for that is quite simple. Of course, with increasing species richness, phylogenetic diversity increases as well. But there is assemblage on the lower hand of the line where we find phylogenetic or functional clustering, means that the species are more closely related than we expect by chance, or we expect by the zero hypothesis here, or we have over-dispersed communities. That means that the species here are more distantly related than just expected by chance. So moving to bats now, there's a very nice study investigating that for bats, and I like very much the main conclusion, which is that one dimension of biodiversity was not a good surrogate for another. So they investigated taxonomic, functional, and phylogenetic diversity, and they simply cannot bring it together in the response to an environmental change, to an environmental gradient. So when moving now to the basic ecological principles behind this pattern, that ultimately leads to the conclusion that we find divergent patterns in phylogenetic and functional diversity if the traits we are looking on are not conserved in the phylogeny. So they have evolved convergently, like here, for instance. So just this small hypothetical tree where the different colors of the species here represent different traits, and we now put some environmental constraint here that filters for a specific trait, we might just get the green ones here to our local assemblage, which is here more or less the box. So this could be one reason for we find more or less phylogenetic functional diversity than we expect. As you see here, the functional diversity is very low because we only have the green species, but the phylogenetic diversity is actually quite high because they assemble from very distantly related clades. But there is a second case that might be true, and that is here, where species which have very different traits here assembled to a local community, but recruited from a very small area of the phylogenetic tree that says that phylogenetic diversity is very low, but functional diversity is high. But of course, environment does not select for the phylogenetic tree or for a specific tree, for a specific clade. The environment selects for species property that allow species persistence in the given environment. So actually, that is what also come out from the discussion about Chelsea's talk. We don't know actually if we measured the correct traits relevant, for example, and that could be the case here. So if there is a trait which is somewhat conserved here in the phylogeny, but we did simply not measure it, we might find this pattern. So actually, this could be the two hypothetical cases that cause a mismatch of functional and phylogenetic diversity. If we put now the gradient here, we would expect some pattern like that. So it is just completely different response of both, and you can see here that is corresponding to here. We have very decreasing functional diversity here because it's select for a specific trait, but increasing phylogenetic trait because we select for species from different lineages, and vice versa here. Okay, enough theory now, moving to real-world data collected in the Bavarian Forest National Park along 48 sampling plots distributed through the whole park area to cover the area. I mean, it's like 20 to 30 kilometers here, where we mount these nice bed quarters for 5 nights per year, pooled them data to get really robust assemblage data for each plot. On each plot, we further mounted a terrestrial laser scanner that sends out million and million laser beams, and is very nice and sophisticated way to get a standardized measurement of vegetation density, which we will use as an environmental gradient later on. And this was combined with an existing database on traits of European bat species published by scientists from the Netherlands. Very comprehensive and very nice scale, and we compiled additionally a phylogenetic tree by mining genetic sequences from GenBank. We are in our pipeline to get most recent information about bat phylogenetic relationships. Okay, and that what's looking like actually just species numbers plot against the vegetation density. You can see with increasing, so that is basically percent of returns. With increasing vegetation density, we get decreasing numbers of species, saying, okay, that's what we expect, that there is somewhat a filtering or somewhat an exclusion of a specific species when moving to denser vegetation. The same is found for the communities, where we just put density of vegetation here. And we always have the elevation as covariate in the models just to correct for different bat activities in lower or in higher, particularly in higher elevation actually. And that matches actually the habitation that we have, like the Beckstein's bat here in the dense vegetation, which is these famous species here adapted to cope with dense vegetation, to pick insects and spiders just from leaves. And on the other hand of the scales, we have like Nyctalus species here, Vespertilio species, which are adapted to fly over the canopy of the forest or hunt in open areas. So actually vegetation, that's just to prove that vegetation density has a massive influence on our communities. So now going to the graph we all want to see is, that is what we find for the functional and phylogenetic diversity. That is an effect size of a null model. And you can see here, this is based on pooling a couple of functional traits, 5 traits in total, and distances from the phylogenetic tree. And that basically matches the first assumption. So the vegetation density seems to select for a specific trait, for a specific species property. But this seems to be present in very different lineages. Just to put it, that is exactly what we hypothesized for the first case, for the A case. So basically that means we have selected the right traits, and that means that there is a trait that has been evolved at very different time. Okay, so far. But when we're going now, because we want to know what's going on, we want to understand this pattern, when we're going now deeper in and pick out single traits, I just picked out 2, I think, for instance, wingspan. So wingspan showed a strong filtering effect along the vegetation density gradient, meaning that assemblages of bed communities in the dense vegetation were composed by mean in smaller species than those of this species, which makes sense because big species cannot fly in dense vegetation. That was what we thought, but actually interesting is that the size of the bed was not the most important factor. What was much more important is actually the ability of a bed to detect objects. So what we see here is quite a nice trait because it has been measured in like experimental setups, putting beds to wind channels. And this is the maximum distance a bed is able to detect an object. What you see here is that this distance decreases, so beds at very dense vegetation are able to detect only very short distance, but with very, very high resolution. By contrast, the open habitat beds, like the Nyctalus beds, they can detect objects like by 70 meters, but very coarse, and that is nothing you need when you want to go in the dense vegetation. So it's more this echolocation stuff that excludes some species than just the morphology of them. And just to sum up, just to come back to one of the first slides where I said that the mandatory stuff for all this is that the traits are convergent and not conserved in the phylogeny. When we just looked here, this echolocation or the good echolocations you can find, for example, here, the Backstein's bed, but also here, the brown long-eared bed, for instance, and they come from, I cut it off here because it comes more here, and they come from very distant related clades. So to conclude, we find strong evidence that a filtering on a specific trait, namely echolocation ability and wingspan and wing loading, is caused the contrasting pattern of bed functional and phylogenetic diversity along the gradient of vegetation density. Thanks.
How long did your scanners detect how much around each scanner?
Yeah, it's between 30 and 50 meters.
Hi, very interesting talk. Do you have any hypothesis why these traits are so labile, not conserved along the phylogeny?
Yeah, maybe probably a reason could be that it was at some time so important to move into the forest because maybe of competition pressure from other bat species that this trait has evolved parallelly and enable bat species to move in dense vegetation just to wend out the competition by other species.
I'm just wondering how heterogeneous the forests are in that area. Like, is there complete sections where it's mostly dense vegetation and other areas that are much more open, or is it pretty spotty across?
Yeah, it's quite heterogeneous because there were a lot of bark beetle outbreaks that create such a mosaic of killed and not killed, half-killed stands. But to overcome this, to standardize somehow, we cut off these laser peaks after I think 25 or so meters, so just to get really the close surrounding of the bed quarter.
I was just curious because I was wondering if there's sort of a scaling issue that's happening here too, because you definitely see a pattern of increased over-dispersion in your traits. So if those traits there's no phylogenetic signal, and then you're getting similar traits in the environment that's just over-dispersed along the phylogeny, right? So if you're getting that effect, you can see it in a number of different plots, but none of the particular plots seem to be actually significant in terms of their effect size. They're all kind of between negative 1.87 and positive 1.87.
Oh no, actually that is just the null model effect size. I didn't show this statistic, but both of these slopes were significant.
Oh yeah, the slope is significant, but the individual plot itself, that effect size is not significant.
No, but the effect size has nothing to do with their significance.
Okay. are you using net relatedness index?
No, completely
sorry,
different.
I was confused then. Okay. So what was the measure that you were
It's a standardized effect size of mean pairwise distance.
Yeah, that's exactly.
From the.
Yeah. So you can ask whether or not that plot assemblage is different from if you were to randomly select the same number of species from an assemblage. But I guess my question is more like if you'd taken all the assemblage of species that were in that particular, in that dense vegetation, and then ran it again, I wonder if that would actually come out as significant.
Okay, you mean the test for the specific, for one plot?
Exactly,
no,
yeah, that's
no.
what I was wondering, if scaling back you'd start to see.
Yeah, no, I didn't look at this. I just smashed the overall effect sizes to mixed models then.
Maybe you guys should go to school.
Yeah.
I want to thank all of our speakers for a really great session. I think the posters are on now. Yeah. Okay. Thank you.